It's a great pleasure to be with you today to share some of our experiences of precision medicine, genomics, and the management of our cancer patients. I'm David Thomas. I'm a medical oncologist, and I head up Omico, Australia's non-profit national precision oncology platform, providing genomic screening for cancer patients right around Australia over the past few years. So that screening program is called the MOST study, or Molecular Screening and Therapeutics, and it's designed to provide genomic screening for cancer patients, but as well to provide access to targeted therapies through clinical trials. So on the next slide, you'll see the mission of Omico to provide molecular screening and therapeutics. Also, we have a program based around personalized risk management aimed at understanding why cancers occur in young people. But we recognize that genomics and cancer care is so important, so critical, that it's essentially a new model of care requiring health systems reform. And finally, we mustn't ever forget that we're a patient-centered program. So this is all about what we can do for our patients. As I said, the Genomic Cancer Medicine program divides into the Molecular Screening and Therapeutic Study, which has a variety of sub-programs focused on areas of priority, such as pancreatic cancer, lung cancer, hematological cancers, as well as being eligible for cancer patients of any origin. From our risk side, we have a study called the Genetic Cancer Risk in the Young study, and we have an intervention program here too, because when we discover that somebody is at an increased risk of cancer, it's important that we have tools that we can use to modify that risk. And in this case, we use whole body magnetic resonance imaging as a screening program to try and detect cancers at an early and curable stage. So what I want to talk to you about today is our MOST study. The concept here is that patients with advanced cancer having molecular profiling can then go on to receive matched therapies based around personalized treatment of targets that are found in their tumor. So initially, when we started this program about 2016, that is about 6 years ago now, we started off within the state of New South Wales, but under the influence of our non-profit entity, over the past 3 years, we've extended that nationally. And now we're open at 23 centres around Australia in every state and territory. And you see the many partners, from academic and clinical partners through industry partners, collaborating organisations. And also we think it's very important that we establish partnerships with similar organisations internationally, because the more that we are able to combine data, the faster we'll make progress. The program has been in extremely high demand. We're actually now at 5,500 patients into the program, and that's well exceeded our 5-year target of 4,095 patients, even though we're only 4 years into the national program. You'll also see that we've seen an extraordinarily broad range of cancers. In fact, sarcomas, the area in which I personally have a professional interest, have been significantly overrepresented in the program, probably partly because options for treatments are limited for rare cancers. But also we have diseases like pancreatic cancer, brain cancer, lung cancer, biliary tract, ovarian cancers, and cancer of unknown primary amongst our top 10 or 12 cancer types. And that's something that we want to see, because we want to see patients who've got limited options, who've got particularly poor outcomes, because it's that population where we expect to make a big difference. After we perform the molecular testing on classical formalin-fixed paraffin-embedded tissue, we analyze that data in our molecular tumor board, and that's shown here in this slide. We get together as a group involving bioinformaticians, clinical team, medical oncologists, pathologists, and cancer genetics experts to review each patient's molecular profile, including asking the question, which are the important mutations here that can be treated? What are the treatment options we can link these patients to? And in some cases, do we see evidence here that might be arising in the germline rather than the tumor, and therefore have implications not just for the patient, but in terms of risk for family relatives who may share that genetic variant that caused this patient to develop a cancer? These are the questions that we preoccupy ourselves with in our molecular tumor board. Our primary purpose, though, is to find treatments for patients, and particularly through clinical trials. In this study, as shown in this slide, we've screened 3,905 patients with a molecular tumor board report. Of those, roughly 798 patients have been referred onto one of our own in-house clinical trials, and another 2,200 odd patients have been referred to treatments outside of our own in-house program through trials that are going on throughout the country. Only about 1/3 of patients have no recommendations at all, although the strength of recommendation varies from what we're calling tier 1 or tier 2 recommendations, which are for treatments that are currently approved on our pharmaceutical benefit scheme or been approved by a major regulatory body internationally, through to tier 3, which have some early clinical evidence, all the way into tier 4, for which the evidence is largely preclinical. And obviously the strength of the recommendation is graduated in that way so as to guide the referring clinician as to which are the strongest possible options for their patient. To the right, you see an example of the benefits of molecular profiling using genomic panels. This is a study in lung cancer. There are 320 patients at this point who had non-small cell lung cancer in the first line. And in the gray bars in this diagram, you see the frequency of detection of these targets using conventional single gene testing, which is standard of care currently in Australia, compared to the results of genomic panels. And what you see, first of all, is that in general, there are more targets identified through genomic panels than there are through standard of care testing, which can be often quite patchy. And secondly, there are certain options for treatments which are really uniquely identified through genomic screening, such as the NRG1 fusions that you see in 5 patients out of this cohort, for which drugs are currently emerging through clinical trials. So comprehensive genomic profiling does seem to offer a benefit compared to standard of care testing, especially in non-small cell lung cancer, a disease for which more than half of all patients will have a treatment target which would be clinically actionable as we would view it. On this slide, we show the rich range of our own in-house trials. We have more than 23 trials that are going on currently, either in-house or through our collaborating partnerships. And these trials are the way in which we maximize the opportunity for patients to receive treatments when those treatments aren't available as standard of care. We think it's important that we do this in the context of clinical trials, both to provide, to avoid out-of-pocket expenses for the patients, but also because it's a way of learning more about what are the best options for patients in the future. And we have a particular trial. I mentioned the aspiration study in non-small cell lung cancer. This trial is specifically designed to answer the question for lung cancer patients, what is the role of CGP and should this be a standard of care? What are the implications for the healthcare system in doing so? And in this program, patients get both standard of care testing and CGP, and I've shown you some of the results of those earlier. And after that, they can go on to a variety of treatments, either through the pharmaceutical benefits scheme as standard of care, or to the right of this panel, receiving treatments which are not yet available as standard of care, but which are available through our clinical trials program. I want to finish off my talk to describe what it means for individual patients. So on this slide, you see a patient with a malignant pheochromocytoma. About 80% of the cancer patients referred onto our program are either rare or less common, which is, again, as I said, something that we're trying to encourage, because we want the patients who have got the least options to be able to access the program. Malignant pheochromocytoma is a very rare cancer, and it's associated with a biomarker, chromogranin A, which in this patient, as the tumor progressed, went from 16,000 6 months prior to referral onto the program to more than 33,000. Unexpectedly, following comprehensive genomic profiling, we identified a druggable target, which is a RET septin-9 fusion gene. Fusion genes are an interesting group of emerging targets, I should say, in general. And there are drugs that target RET. And as a consequence of this genomic profiling, this patient was able to access the RET inhibitor selpercatinib. And you see to the right the dramatic PET and objective CT-based responses to this drug, this patient receiving that drug. You see the chromogranin A levels fell from 33,000 to 338. And that was purely because this unexpected target was identified thanks to access to molecular profiling. And this is a second patient, which illustrates the point about germline findings. We had a patient with an endometrial cancer, metastatic endometrial cancer, who unexpectedly turned up a germline PALB2 variant. And PALB2 is associated with an increased risk of breast and ovarian cancer. It belongs to the same family as BRCA1 and BRCA2. these genes are genes for which the PARP inhibitors appear to be an effective targeted therapy. And in this patient who had an endometrial cancer, the identification of the PALB2 gene led to her being able to access a PARP inhibitor, and that was associated with a response to treatment for this patient with incurable cancer. The general, this is written up in a case report, which you can get access to as outlined here. And the general point of this, this single case was that there's a need to ensure that genetic testing is applied widely over a broad range of tumor types, not only for the benefit of patients for whom the result may directly inform their cancer management, but also for their families in whom appropriate institution of timely genetic counseling has the potential to save lives. And that's a very important message about the potential of genomics to impact upon our patients and their family members. And that potential is what we're trying to unlock here in Australia through programs like MOST. Thank you.